So I think this is your first lecture of science and technology. Am I correct? Okay. So before we begin the discussion of the science and technology section, first we'll have the brief overview of this particular segment of general science and technology portion. Now, how this, first of all, this is in the mains, it is GS paper 3, the science and technology portion. But there is some overlapping in terms of environmental science and technology. So you may get question in other papers also sometimes. So general science and technology. So you will find in the syllabus of prelims exam as general science, and in the mains you will find science and technology chapters. But in the question paper you will see that 90% of questions of prelims, they are asked from the science and technology syllabus. So both are inseparable. So both are what? Inseparable from each other. This is the key takeaway. So definitely you need the foundation of general science. General science means your basic understanding of the chapters of physics, chemistry and the biology as a part of the general science. Here, just like other general studies paper, you have to begin with the NCERT textbook with the 9th and the 10th standard. Now physics, chemistry, 9th and 10th standard. Biology, apart from 9th, 10th, you also need to refer to the 11th and the 12th standard NCERT textbook. So this is with reference to the NCERT textbook. Then as we see the syllabus of, we will see the discussion on the syllabus of science and technology. So that will give you the roadmap that how we have to proceed in science and technology segment. First I will write the references. So one reference we have understood, the NCERT textbook. So NCERT 9, 10, physics, chemistry, biology apart from 9, 10, 11th and 12th standard also. Now the other references apart from NCERT textbook. So first one was the NCERT and the second one, right? Now it is highly multidisciplinary and interdisciplinary approach in case of the science segment. So you won't find a particular textbook unlike history, geography or the polity to be referred here. But definitely current affairs will play a significant role in framing questions. So the relevance, the context of asking question will be overlapping with your current affairs part. And current affairs means the duration is important. So duration here will be, for example, the next year prelims exam. So in which month the next year prelims exam is there? In the month of May. So your preparation starts from May 2022 till April, in fact 30th April 2023. So 1st May 2022. So this will be the duration. This is what the duration which will be looked into by examiner to frame the questions related to what? Current affairs. So for this duration, the aspirants, those who are appearing for this exam, will be accountable to answer any question related to this duration. So you can see you are already now in the month of October. So what about the May, June, July, August, September? Is it clear from where questions will be framed? Now current affairs reference, as it has already been communicated to you. So this is related to the newspaper reading. Which includes both the Hindu and Indian Express. And newspaper reading will be as such, just like without the preamble, you are not reading the Constitution of India, isn't it? So this newspaper will become the preamble here, and then you have to read further. Is it clear? So that is why newspaper reading, and each day newspaper reading will be a strategic task for you, because that will form the strategy for the examiner also, for aspirant also, to put the question and also answer the question. So therefore you can understand the context is current affairs appearing for next year prelims exam. Is it clear? So you have to understand this duration is important, and this will be only from the newspaper reading. There cannot be any magazine, any source that is going to give you each day newspaper reading material in comprehensive. There may be some addition, deletion. So the one who is making the current affairs magazine or other resources that you want to refer, it cannot replace the newspaper reading, because he or she cannot know all the multiple dimensions of the chapters which could be important for examiner and can be picked up. So this is the most important resource for your reference. So it is highly dynamic and evolving, this portion, science and technology portion. Because basic concept also will be related to. So newspaper reading will pick up the current affairs statement, but related to that the basic concept that are required. So that will be also in the multiple statements of prelims exam. So even your basic concept also that you are going to clear, it should be in the context of the newspaper reading only. Then the next one is the third one, current affairs magazine. One is the down to earth current affairs magazine that you can refer. Then from institute portal, you will get each day, isn't it? The web portal of the institute will get the current affairs news. Fourth, right, the important websites. WHO website, website of NASA, website of ISRO. So these are important websites. So whenever there is some factual challenge before you, immediately you refer to the concerned website, the parent website. So international public health concern will be there. You will refer to what? WHO website. If any news related to the launches, international space missions, you refer to the NASA website. Then India's National Space Agency ISRO website. Then next one is Ministry of Health and Family Welfare website. Ministry of Environment and Forest and Climate Change. Ministry of Environment, Forest and Climate Change. So these are not exhaustive, but definitely these are important websites to be referred. Sometimes you may also require to refer Ministry of External Affairs website also. Ministry of External Affairs website, because there are certain global collaborations in science and technology between India and other countries. Bilateral, multilateral also, you can refer directly to the Ministry of External Affairs website. Then the fifth one, right, the latest budget and economic survey. So the latest budget economic survey before your prelims exam. So budget, that is as a policy tool. Budget is what? This is a policy tool of government. So here you will refer to what? What are the policies regarding the energy security? So policy tool of government of India, isn't it? So what about energy security? Then renewable energy, information technology, robotics, public health, agriculture, is it clear? So government will announce along with the budget allocation of resources that what are the priority areas in these specific areas related to directly, indirectly, the science, technology, defense and internal security, cyber security, see in what context you have to approach the economic survey and the budget, how you have to decode the budget, decode the economic survey. Climate change, we'll see more chapters. For example, another chapter that he will mention here is biotechnology. Biotechnology also. So one is in the newspaper that you will read the analysis part of the current affairs related to these chapters or the portions along with the focus of government. Both newspaper are essential, either to both the newspaper. If you want, you can go for third also, Times of India. Fourth also, Business Line. Fifth also, Economic Times. Is it clear? But at least these two. Because every newspaper will come out with the diverse viewpoints. Because it is not about clearing prelims or mains. When you are going to face the interview, then you need to know the diverse viewpoints about that topic, the pros and cons, everything. And that will give you only the newspaper reading. Is it clear? So you have to understand the context of this exam, what is the need of examiner that wants to focus, which person should be selected. Is it clear? It is not about this person is only fit who will be selected, but the temperament also will be checked into in the personality development in interview. A person may be fit in terms of prelims main, but not may be fit in terms of induction into the services based on interview. So they will require innovative solutions. Problems everybody knows, but what is the innovative solution that one comes out in these areas? That will be looked into by the examiner. So this was the fifth part. Now write the sixth one. Sixth reference. Now sixth reference we will write here. Nobel Prizes in physics, chemistry and medicine. Last 5 years, including the the current year, which is yet to be announced. After 1 or 2 days, you will now start reading the newspaper with respect to the Nobel Prize also. Seventh, right, previous years prelims and mains question papers for civil services examination. So this will provide an insight that what is the depth, extent to which examiner can go on a particular topic to ask the question, isn't it? Sometimes the topic remains the same, but the framing of questions may change, the concept remains the same, but you need to know how the examiner writes the statement of the questions in prelims and mains, so that tomorrow when you're reading newspaper, then you can focus on the spirit of that particular question. So these are the important references for our science and technology portion. Now the next one is, what is the syllabus defined for the science and technology? So syllabus of science and technology. Already, as we discussed in the beginning, that 90% of questions prelims will be asked from the syllabus of science and technology. So indirectly we are discussing both. So the first one is biotechnology and issues related to intellectual property rights. IPR issues, which includes patent, copyright, trademark and geographical indication, GI, now the biotechnology chapter, there will be overlapping with the public health portion because it provides solution to the issues of public health. So biotechnology tool or technique, we call it as the genetic engineering, also known as recombinant DNA technology, so the tool of biotechnology is called genetic engineering, also known as what? Recombinant DNA technology. So by using this genetic engineering, we develop whatever products that you develop by using this. For example, vaccine. So vaccines develop by using genetic engineering. As I told you that there will be overlapping with the public health portion. So in public health, here we will discuss the basic concept related to what is a vaccine, what is a vaccine, how vaccine works, isn't it? Then comes the types of vaccine, types of vaccine. So how vaccine works, types of vaccine, what are the principles behind vaccine development. Now, so this is an innovative solution, but what is the issue? Issue is certain life-threatening disease. So public health international emergency, whenever there is some public health international emergency, then we have to be proactive in approach. This public health international emergency in the form of global spread, crossing national international boundaries, increasing morbidity, mortality in the society. So it may be declared as a pandemic, declared as a pandemic, the global spread of a disease. For example, the current COVID-19 pandemic. So there were large scale implications, impact on the society, social impact, economic impact, impact on the health of individual society. So there was complete disruption of society due to this COVID-19 pandemic. So every individual in any part of the world was looking for solutions for this pandemic, isn't it? So when we look back the year 2020, compare with 2021 and now in 2022, there is a huge difference in these 2 years. We have seen that nightmare of COVID-19 pandemic and mass restrictions also. But now there is complete ease of restriction, isn't it? So one is the basic understanding that the pathogen means what? Disease causing organism. So pathogen for this COVID-19 pandemic, that is the SARS-CoV-2, isn't it? Which we write as severe, acute, respiratory syndrome, coronavirus 2. So one is the basic details of this virus that is responsible for the COVID-19 pandemic. Now, generally a vaccine development, it takes 10 to 15 years. It is a highly complex process. There are many health indicators that we have to gather in the course of development of a vaccine. But in a pandemic-like situation, we have expedited, based on medical advancement approvals, we have expedited the introduction of this COVID-19 vaccine. If you consider regarding the vaccine development to control this pandemic in Indian context, so vaccine against COVID-19 that India has used to control this pandemic. If this COVID-19 is the most important event of this entire century, or maybe in your entire life, isn't it? So to address this issue, what are the different vaccines that India has used? One is the Covishield. Another one is what? Covaxin. Now later on other vaccines also were introduced, but these are the two primary vaccines, isn't it? Now under types of vaccine, we will discuss types of vaccine, the different types. Viral vector vaccine, mRNA vaccine, DNA vaccine, peptide vaccine. These are important types in terms of the vaccine. So one statement in the prelims exam could be that which Indian manufacturer, what is the name of the vaccine and name of the manufacturer who introduced the vaccine. So for example, Covaxin. So Covaxin introduced by which manufacturer in India? Bharat Biotech. Bharat Biotech. So manufacturer is the Indian manufacturer, indigenous. Indigenous means what? Homemade, isn't it? So Bharat Biotech, Indian manufacturer, developed this Covaxin, which is a COVID-19 vaccine. So this is a factual statement, isn't it? That everyone should know, because this is what? Indigenous vaccine. But the second statement will be which type of vaccine. So what type of vaccine out of these? What is your answer? This is how questions are asked in the exam. These are all related to what? It connects our life immediately, because the most important event of this century is COVID-19 pandemic. Solution to this is what? The vaccine. Which vaccine used in India? Covishield, Covaxin. So you have to know basic details. One is the factual statement. Which Indian manufacturer developed this vaccine? Name of the vaccine. But the next statement will be type of vaccine. So what you will select out of these four? Answer is none of these. Answer is what? None of these. So this comes under inactivated vaccine, because it contains non-replicating or inactivated SARS-CoV-2 virus or pathogen. So it is what? Inactivated vaccine. Inactivated vaccine means where the pathogen means what? It could be bacteria, it could be virus, against which your body needs to develop protection. So the pathogen used there, the virus used there, is non-replicating. Non-replicating means the vaccine virus will not multiply in your body to ensure safety of the vaccine. So inactivated means what? Non-replicating, non-multiplying. The vaccine virus, when injected into the human body, will not multiply in the human body. So that safety has been ensured in development of the vaccine. So this is how an example of a topic and how questions can be asked related to this public health. Is it clear now? So this topic on public health is very important, where we will discuss about important diseases impacting the society. What are the important diseases? Once you know the disease, then comes the solution. See, I don't have remote to control the temperature of AC. That you have to look at the. So diseases includes HIV/AIDS, malaria, HIV, malaria, dengue virus disease, Zika virus disease. Then you must have read about tuberculosis, there are thousands and thousands of diseases, but which disease, which is in current affairs, which disease impacts the society, that is important for the examiner and also important for the exam. Polio virus, Nipah virus disease, Ebola virus disease, hepatitis, A, B, C, D, and the E. So there are 5 different types of viral hepatitis. So these are what? Viral hepatitis. Viral hepatitis. So this is also important for the exam. Now, apart from these diseases, some of the diseases which are in current affairs, for example, you may have read a lot on the monkeypox virus. Then recently you read a lot on the lumpy skin disease. Now there is a term known as related to disease, vector-borne disease. So vector-borne disease means there is a carrier to transmit the infection. There is a carrier to transmit the infection. For example, mosquitoes, insects, so they can act as carrier to transmit the infection. So example under vector-borne disease like malaria, dengue virus disease, Zika virus disease. These are important examples of what? Vector-borne diseases. Now, what about lumpy skin disease? Is it a vector-borne disease?
No. Insect, yeah, it's
Ticks and mites. The lumpy skin disease also comes under what? Vector-borne disease. But this is affecting, then there is a term known as zoonotic disease, which means animal to human transmission, animal to human transmission. Animal to human transmission. For example, bird flu, swine flu. Is it clear? They come under what? Zoonotic disease. Both belong to influenza virus. Both belong to what? Influenza virus. Yes, you can write COVID also. COVID-19 also zoonotic. Coronavirus also zoonotic. So these are some examples. Now, what about lumpy skin disease? Is it a zoonotic disease? Now, lumpy skin disease that affects mainly whom? The cattle. So there is cattle to cattle transmission. Is it clear? In lumpy skin disease. It is caused by a virus, lumpy skin disease. But this is not transmitted from cattle to human. So there is no animal to human transmission yet noticed for lumpy skin disease. So it is not a zoonotic disease. So it is a vector-borne disease, but it is not a zoonotic disease. Is it clear? So when you are reading newspaper, you have to be careful what you are reading and what you are making notes from the newspaper reading. Because these are related to what? Current affairs. So therefore, I have given some examples. Then under different types of vaccine that you have written like viral vector, mRNA, DNA, peptide vaccine. one of the greatest breakthrough of this century, which will provide various solutions to diseases or future pandemics, is nothing but the importance given to the mRNA vaccine. So our special focus also will be in the course of discussion on mRNA vaccine. What is the current research and development blueprint for the mRNA? What are the various solutions it can provide to the society? So you have to understand where is the focus of examiner. In that context, you have to read and revise. That is the most important part, because there is no defined syllabus here. So this is in brief about the related to public health. Then public health, here we are also going to discuss on concepts related to nutrition, concept related to immunity, so nutrition particularly will focus on the different types of vitamins and their deficiency. So vitamins and their deficiency. So immunity means basically the role of white blood cells, So how our natural immunity tries to protect us from pathogen?
Multiply numbers.
And then we'll discuss on the vaccine induced immunity. One is your natural immunity, how it works. Then we'll discuss on the vaccine induced immunity also, as a part of the immunity only. So these two are important portions, comprehensive portion related to the public health, apart from the disease aspect that just now I have written. So disease is there, nutrition, immunity, all these are interrelated. Now write the second one. So after biotechnology, public health, the next important chapter write information technology. Now information technology, there is a special focus nowadays on I 4.0. So what is this I 4.0? Fourth industrial revolution. In history you read about the first, second industrial revolution. Here we are going to focus on the fourth industrial revolution. Now what are the components of fourth industrial revolution, which will overlap with the discussion of the IT? Another chapter is the robotics also. So this fourth industrial revolution includes discussion on robot or drones, Once you write down what are the components of I 4.2. So robot or drone, artificial intelligence, now branch of AI we will discuss in the name of machine learning. Then write IoT, Internet of Things. So now more and more devices, they are getting connected to network or the internet, and we are extending our communication to all the devices in the network. So we are now gathering data by connecting the device to the internet, refrigerator, washing machine, your vehicles, everything is getting connected in the ecosystem of IoT. 5G, LTE. So fifth generation digital wireless mobile communication or long-term evolution. So revolution, economic field revolution or social. So how we can increase accessibility and bridge the gap between rural and urban divide. So that could be only possible with the help of what? Extending those advanced healthcare services to the from urban to the rural areas by using the 5G connectivity. Then it also includes blockchain technology. It also includes augmented reality AR and virtual reality It also includes cryptocurrency, and the combination of all these together metaverse. So the platform which will help converge, all these technology on one platform, that platform will be known as what? Metaverse. So convergence of all these digital technologies on one platform, that becomes the metaverse. Now, every industrial revolution is characterized by a certain outcome. For example, what was the outcome of first industrial revolution? Steam engine. Second one was mass production using electrical energy. Third one, IT. And the fourth one is what? Yes. What is the outcome of I 4.0, fourth industrial revolution? Cyber physical systems. Cyber physical systems. Is it clear? So every industrial revolution is characterized by a certain unique outcome. For I 4.0, what is the outcome? Cyber physical system. Example of cyber physical system: autonomous drones, autonomous vehicles. Examples of what? Cyber physical system. Third chapter you can write, which is mentioned in your syllabus, robotics and artificial intelligence. So you can see there is overlapping of IT as a part of I 4.0. So third chapter is what? Robotics and artificial intelligence. Now currently we are in the era of big data. We are in the era of what? Big data. So this big data, it includes increase in volume of the data, increase in variety of the data, and also increase in what? Velocity. So real time generation of the data. So this data could be text data, audio, video data. It could be personal data, your social data. All these are part of the big data. And how we are capturing this big data? What is the tool to capture this big data? Now, data itself cannot do anything. Data itself, it cannot take any decision. So first we have to acquire the data, store the data, process the data, and that processed data is subjected to certain decision making. So big data, this is subjected to data analytics. So when you are converting data into knowledge for decision making, then you are using what? Data analytics. And the tool for data analytics, that is known as artificial intelligence. Tool of data analytics will be known as what? Artificial intelligence, which is basically an algorithm. It is an what? Algorithm or set of instructions. Algorithm or set of instructions or some software program which has decided that what action it is going to take based on the data given in that context. So context of decision making, context of action will be decided based on what? Artificial intelligence algorithm. This will decide. And who are using this on large scale? So globally, it is the big tech companies which are relying on the big data and data analytics, including the AI. So including your Facebook, WhatsApp, Instagram, all these three are the same, isn't it? Then another one is your Google. So when you are using Google Map, when you are watching YouTube content or videos, or when you are using Google as a search engine. So basically what is happening that, for example, a very common understanding you can see in day-to-day life, when you are adding someone as a part of mobile directory in your WhatsApp directory in your mobile phone, then tomorrow you will find the same individual reflecting in your Facebook under the column people you may know. But the question is, how do you know? Isn't it? So don't you think it is using some algorithm that this individual, since it's part of WhatsApp, may be known to this individual? So that is an algorithm which has pulled up all those individuals who are part of WhatsApp now, bringing them on a platform of people you may know. Then it is up to you. You are giving the request or deleting it, isn't it? You have to take your own decision, isn't it? So this is how they are using what? Artificial intelligence only. So this company's business model runs on artificial intelligence completely. Similarly, when you are on Google platform, whether you are browsing certain websites, so that will be recorded. Whether you are watching certain videos over the YouTube for the past so many years, getting recorded. When you are on Google Map, it is getting recorded, which place you are visiting, how many times you have visited, in between what direction you have selected, the path to reach the destination, in between where you have stopped. All these are your personal data related to your privacy, recorded by the Google, isn't it? So this indicates there is increased surveillance. We are under increased surveillance, which will automatically compromise your privacy. For which we want to evolve our data protection laws, isn't it? Every country is trying to protect its own citizen data, because all this data is going and getting collected where? At the server located in the country to which they belong. So therefore we are advocating data localization in India only. Then this big tech also includes Amazon, Apple, Microsoft. So all these are part of what? The big tech. So you have to understand the context of discussion, what you are reading in newspaper, how it is important for you, and how it is connected to your syllabus that you are writing now, one by one. So these three V's together, isn't it? These three V's, they together form what? The big data. So increase volume of the data, variety, the different context to gather the data, and then real-time access of this data. All these three together form what? The big data. And the fourth one will be the veracity, the truthfulness related to the data. Currently, you just know about the three V's. We'll discuss when we come to this topic. Now, the fourth one, right? Nanoscience and nanotechnology. Nanoscience and nanotechnology. So this relates to significance of nanoscale, and nanoscale that relates to, under bracket write 1 nanometer to 100 nanometer. Then role of nanomaterial, including graphene and carbon nanotube. So nanotechnology, nanoscience and nanotechnology, one is the significance of nanoscale and the role of nanomaterials, graphene and carbon nanotube. Then the fifth chapter, space science and technology. This is also very important chapter, space science and technology. So here we'll focus on what? Role of ISRO. So different organizational setup of ISRO, different divisions of ISRO, and what is their exact role. Then types of satellite, types of orbit, types of launch vehicle or rocket, so types of satellite, types of orbit, types of launch vehicle or rocket. Now this chapter, it also includes, although that could be another means portion of discussion related to the astrophysics. So astrophysics, it deals with the study of stars and galaxies, the galaxies. So basically our focus will be on cosmology, that is the study of universe, celestial objects, so objects in the sky, that is the celestial object. which includes your solar system, planets, including sun, and also the stars, all are part of the discussion. Now, to gather data about the cosmos, we have to take the help of what? The space telescope, isn't it? So how do you gather data about these celestial objects? You have to take the help of space telescope. For deep space probe, now to go distant in the deep space by using the space telescope. One of them you already know, the Hubble Space Telescope. And currently you are reading on James Webb Space Telescope. So what is the principle underlying deep space probe? How do you reach in the distant location in the space to gather the data? So questions can be from the space telescope also sometime. In the prelims also, mains also. But again, as I told you, current affairs, that will be the very important portion to set up the relevance, context in which examiner should focus the question in prelims and the mains exam. So to provide better resolution, clarity of image, data, isn't it? All these modifications will be needed in the space telescope. So that was the 5. Now write the 6th one. Indigenization of technology. Indigenization of technology. This indigenization of technology, the focus is on what? India's own research and development, innovation, in the field of space, in the field of defense, in the field of IT, in the field of health and medicine, or biotechnology, in the field of climate change, in the field of agriculture, in the field of AI, so the special focus of government is on what? To make India self-reliant. Self-reliant indicates here what? The principle of that Atmanirbhar Bharat concept. So that is reflected in this chapter of indigenization of technology. Is it clear? So this is completely dynamic and related to the current affairs. So what we discuss here, that the principle of indigenization or Atmanirbhar Bharat, that is a special focus of government of India. So how India is innovating in the field of artificial intelligence, in the field of space technology, in the field of defense, agriculture, in the field of climate change, isn't it? Or in the field of vaccines. So that is the 6th one. And the 7th one is contribution of Indian scientists. Contribution of Indian scientist. So one is the Indian scientists who have been already awarded Nobel Prize in those physics, chemistry, medicine. Another one, so whoever Indian scientists awarded the, so you are asking about what is the meaning of indigenization. As I told you, homemade. Is it clear? This is the meaning of indigenization of technology. For example, just now I have written about co-vaccine. So co-vaccine is what? Totally indigenous. Covishield, that was in collaboration with what? Oxford University and AstraZeneca of Serum Institute of India. But co-vaccine was 100% indigenous, homemade vaccine. Is it clear? That is just an example. So these are the important chapters in your syllabus from where questions can be asked in prelims also, mains also, in the interview also. So you have to remain vigilant while reading the newspaper. That is the most authentic source, relevant source while reading newspaper and writing the notes from them. So this is about the roadmap action plan for the science and technology segment. When we are going to discuss one by one, then I will show you some of the questions also that have come in the mains exam, that have already come in the prelims exam, so that you are able to connect yourself while the discussion of these topics. So the today's discussion in the class, we are going to begin with the public health portion, because that requires a lot of basic concept in public health. And public health, we are going to begin the discussion with the term immunity. Yes. No. After me now, some more teachers will be joining to cover the portions that have been listed here. Now, immunity means ability of the body. Write down ability of the body. Immunity is the ability of the body to fight against the pathogen under bracket write disease causing organism it involves the role of mainly the role of white blood cells, now before we discuss the role of white blood cells, write the heading composition of blood. Write the heading composition of blood. So under composition of blood, we can see here 55% proportion is the blood plasma and 45% are the blood cells. Now write composition of plasma. In the composition of plasma, we can write here 92% is water only, and the rest 8% of plasma includes plasma protein, nutrients, gases, and the minerals. The blood cells includes erythrocyte, leukocyte, and the thrombocyte, so the scientific name of red blood cell, that is erythrocyte, for white blood cell leukocyte, and for the blood platelets it is thrombocyte. These are the scientific names: erythrocyte for red blood cell, leukocyte for white blood cell, and the thrombocyte for blood platelets. Now, examples of plasma protein. First one, prothrombin. Second, fibrinogen. Third, globulin. So these are some important examples of plasma protein. The first two, that is prothrombin, fibrinogen, the first two. They are together known as blood clotting protein or factor. So the prothrombin, fibrinogen as a part of plasma protein, they come under what? Blood clotting protein or factor. Globulin is used to synthesize So globulin is used to synthesize immunoglobulin, also known as antibody. So globulin is the raw material. Globulin is a plasma protein. This plasma protein, globulin, is the raw material to form antibody in your body. So in this entire pandemic, the term antibody has become a celebrity. Everyone speaks on what? Antibody. But this antibody is produced in your body by using the raw material known as what? Globulin, which is what? A plasma protein. Now, how antibody protects you against the virus, isn't it? Against the virus infection or disease, that we will follow the discussion. But as this globulin that helps in formation of antibody in your body, an antibody protects you against infections. So it provides you immunity, resistance against disease, isn't it? So that is the reason why antibody is also known as what? Immunoglobulin. So this globulin is used to provide something that gives you immunity. That is the antibody. So it is also called immunoglobulin, represented by the symbol capital A, small g. And this antibody can be represented by the symbol capital A, small b, the symbol of antibody. Now, as nutrients, you can write example as glucose, amino acid, so blood is a transporting medium. Blood is what? A transporting medium. Blood transports glucose, blood transports amino acid. Amino acid, these are building block of proteins. These are what? Building block of proteins. Then gases, carbon dioxide, oxygen, example of gases. Minerals, Na sodium, K potassium, Ca calcium, these are examples of minerals. Na sodium, K potassium, Ca calcium are examples of minerals. So blood is actually a fluid connective tissue. Fluid connective tissue. The fluid part of the blood is the plasma, and in the fluid plasma are suspended the blood cells, including the erythrocyte red blood cell, leukocyte white blood cell, and the thrombocyte blood platelets. Now, based on this basic composition of the blood, now we will see the production of the blood cells. So, what is the site of production or formation of blood cells? Which one? Yes, pelvis means hip bone. Platelets and prothrombin related. Platelets, these are the blood cell, and fibrinogen, prothrombin, these are blood clotting proteins. So now we can see here the site of formation of blood cells. Now the production or formation of blood cells, is it clear? We are discussing on the site, but before that, formation of blood cells or production of blood cells. That is known as hematopoiesis. Is it clear? So formation of blood cells, production of blood cells is known by the term hematopoiesis. Bone marrow is there. In the entire bone, there is bone marrow only. Is it clear? Bone marrow means hollow interior of bones. Is it clear? It is at the ends of long bone, the presence of red bone marrow. We say that it is at the ends of long bones, there is presence of blood stem cell. And that is the major location site for the production of new blood cells. Ends of long bones. So, one prominent location is a hollow interior of bone is called bone marrow. Within the bone marrow, the location of blood stem cell, that becomes the red bone marrow area. So, within the red bone marrow area, we have what? The blood stem cell, also known as what? Hematopoietic stem cell. So, from these blood stem cells, continuously, new blood cells are produced in your body, depending upon the lifespan of that particular blood cell. So from this blood stem cell, RBC is produced, new white blood cells are produced, and also the blood platelets. So all the blood cells are formed where? In the red bone marrow area. Is it clear? Because of the presence of what? Blood stem cell. As it the process is hematopoiesis, blood stem cell also known as what? Hematopoietic stem cell. So regarding the site, so one prominent site is your hip bone or pelvis for the presence of red bone marrow, where the new blood cells are produced in your body. Another important location drawn here is the ends of long bones, like your thigh bone or the femur. Then another important location will be your vertebra. So vertebra means individual backbone. Individual backbone. Is it clear? So vertebra, plural is vertebrae, or backbone, also known as spine. So individual backbone, individual vertebra, that also contains red bone marrow and blood stem cell, and that is also important location of production of new blood cells in your body. Is it clear? So these are the some important locations or site for the production of new blood cells. So whenever we are donating blood to a blood bank, isn't it? Or to the recipient, the new blood cells are continuously produced, isn't it? So it is not going to exhaust in your body. But it is not that every day you can donate the blood. There is a certain time gap, isn't it? Depending upon the lifespan of the production of the new blood cells. So generally 120 days, the time gap for donating blood to the blood bank. So to blood bank, you can donate the blood after 120 days. Now, before we proceed further, when we are using the term stem cell, just a brief introduction to the stem cell. In more that is elaborate form, it will be discussed in the biotechnology segment, because this is a part of the biotechnology. Now, stem cell, this is derived from the word stemness. So the term stem cell derived from which word? Stemness. Stemness means to originate. So the term stemness means what? To originate. Now all the cells of the body originate from where? The stem cell. Is it clear? All the cells are derived from stem cell only. So that is why the term we are using what? The stem cell. So stem cell, wherever present in your body, that part of the body can regenerate, repair continuously. Because stem cells, they are known for two fundamental property of a stem cell. One is the cell division, which is continuously in case of the stem cell. So due to continuous cell division, it provides you unlimited source of new cells. It provides you what? Unlimited source of new cells because of cell division. And that becomes what? Self renewal, self renewal. Is it clear? So stem cell, they continuously divide through cell division, and due to which they provide you unlimited source of new cells. So that is meaning the term self renewal property of stem cell. This is one important fundamental property. Second important fundamental property is differentiation. Second important fundamental property is what? Differentiation. Differentiation here means that it is going to differentiate into a specialized or special type of cell and going to perform the specific function in your body. So you can say as functional specialization. Is it clear? Functional specialization. Because as you know that the red blood cell role is what? So red blood cell, white blood cell, all are produced from where? The blood stem cell only. But the red blood cell role is what? To transport oxygen. But the function of white blood cell is to fight against bacteria and the virus, isn't it? And blood platelets, they help in blood clotting. So they are derived from the stem cell only, but they have different functions. That is known as what? Differentiation. So differentiating to perform a specific task or function for your body as a functional specialization. So these are the two fundamental property. One is cell division, another one is differentiation or functional specialization to perform a specific task for our survival of the body. So wherever these stem cells are present in your body, that part of the body will show continuous regeneration and repair. Is it clear? Wherever they are not present, that part of the body cannot regenerate. For example, when there is death of liver cell, new liver cells can regenerate. But when there is death of heart muscle cell in heart attack, then it cannot regenerate. This is the difference. That wherever the stem cell is present in your body, continuous production, supply of new cells. Wherever it has exhausted the stem cell, there is no supply of new cells in that part of the body. So the blood cells that we discussed, one is the red blood cell, lifespan 120 days. Can nerve cells regenerate? No. If there is spinal cord injury, then it cannot regenerate. Microliter. This is the symbol of microliter, 1 μl. And 1 microliter is equal to 10 to the power minus 6 liter. So we are considering the concentration of blood cells in blood examination in about 1 drop of blood in a healthy person. RBC count should be 5 million per microliter of blood. Platelet count, all of you know, in day to day nowadays, the number of dengue cases are rising where platelet count decreases. So that is 1.5 lakh to what? 4.5 lakh per microliter of blood. So these new blood cells are continuously produced in your body after a time gap of what? 120 days for RBC, after a time gap of 10 days for blood platelet, and about a week for the white blood cells. So there is continuous regeneration of new blood cells. So this is the reason we discussed. To donate blood to the blood bank, you have to wait for 120 days, 4 months. μl, as I told you, this is a symbol of microliter. Microliter means 1 into 10 to the power minus 6 liter. So that is the volume that has been perceivable limit. So we go for 1 unit for 1 individual. Now that is around 100 milli. What is? See, whenever there is injury to the body, in case of injury, there could be blood loss or bleeding. Now, excessive bleeding could be fatal for the individual. Body has to maintain blood volume. So, body initiates a self-defense mechanism called blood clotting. In blood clotting, there are raw materials which are needed, formation of blood clot. These raw materials are known as blood clotting proteins or blood clotting factors. Just now I have written what? Prothrombin, fibrinogen. In case of what? Blood clotting proteins. So, whenever there is some injury to the body, to initiate the process of blood clotting, the blood platelets, as you can see here. So, blood platelets after the injury, they release chemical signal to initiate the process of what? Blood clotting. After this chemical signal released, it will start involving blood clotting proteins to form the blood clot, including prothrombin and fibrinogen. So recruitment of, involvement with what? Recruitment of prothrombin, fibrinogen to form the blood clot requires the activation signals coming from where? Blood platelets. Is it clear? And formation of blood clot requires multiple factors. One is the blood clotting protein. Blood clotting also requires vitamin K also, because vitamin K will be needed by the liver cell to produce this prothrombin and fibrinogen. So this prothrombin, fibrinogen, where they are produced? They are produced in your liver. Is it clear? Now liver cells to produce them requires the input factor known as vitamin K. If liver is the factory to produce blood clotting protein, prothrombin, fibrinogen, that factory requires a raw material input factor in the name of vitamin K. Is it clear now? Then, in the process of blood clotting, our body also requires calcium mineral. Calcium is also essential for blood clotting. So, any one of them which is deficient in your body, so that can delay the process of what? Clotting, and can result in excessive bleeding. Yes. Nerves. No, it is the injured platelets. They send the chemical signal called thromboplastin. Is it clear? Not the nerves. When we come to the white blood cell, there I will tell you the primary and secondary lymphoid organ. Is it clear? Till now we have just discussed about the basic composition and the types. How platelets important in dengue recovery. So dengue virus suppresses the bone marrow region from where blood platelets are produced. Therefore, the blood platelet count decreases in dengue virus disease. So this is the basic composition of the blood, and these are the blood cells that we discussed. So role of red blood cell is transportation of oxygen. The most important role of red blood cell. So you can see that this is our red blood cell produced from the bone marrow region, from the blood stem cell, lifespan 120 days. So this red blood cell, these are what? Biconcave. They have a disc-like shape. So they have a disc-like shape, you can see. In the middle, that is two depression. There are two depression in the RBC. That gives the name biconcave RBC. Then non-nucleated means there is absence of nucleus in the red blood cell. Now inside red blood cell, you can see the presence of what? Oxygen carrying iron containing protein known as hemoglobin. So hemoglobin is chemically a protein, and it contains what? Which mineral? Iron. Oxygen combines with the hemoglobin, oxygen that you are breathing combines with the hemoglobin, and then it is transported to different body cells. So red blood cell, to accommodate hemoglobin and to maintain oxygen carrying capacity, it has eliminated nucleus. So the absence of nucleus helps accommodate by increasing the surface area to accommodate what? Hemoglobin. So absence of nucleus is an advantage. How it is advantage? It increases the surface area to accommodate hemoglobin and maintain the oxygen carrying capacity of the red blood cell. Surface area to accommodate hemoglobin and increase the oxygen carrying capacity. Now we have written that non-nucleated. So what is the rationale behind RBC becoming non-nucleated? Why nucleus is absent in red blood cell? So that it can increase the surface area to accommodate hemoglobin and maintain the oxygen carrying capacity required by the body. So absence of nucleus is an adaptation in our body so that it can maintain the oxygen carrying capacity. Yes, your RBC is surviving 120 days. Is it clear? And then we are surviving. If RBC survive, then only we can survive. No, this nucleus is eliminated. There is no DNA in the RBC as there is no nucleus. You cannot use RBC for DNA fingerprinting. Is it clear? You can use the white blood cell for DNA fingerprinting, not the red blood cell. Now, biconcave nature of RBC, it provides flexibility to your red blood cell. So biconcave also has significance. What is the significance? It increases flexibility in the movement of RBC. Flexibility in the movement of RBC, that it can change its shape. So RBC can change its shape. It can squeeze itself because of biconcave nature. If the passage is narrow, then RBC can squeeze itself to squeeze its size, and then it can make its way forward. For example, blood vessel. So blood vessel provides the passage to the RBC to move forward, isn't it? So this indicates what? The blood flow inside your blood vessel. Now all blood vessels are not of uniform diameter. Blood vessels, they may branch into very tiny blood vessels, and tiniest blood vessel, thinnest blood vessel, that is known as what? Capillary. So thinnest blood vessel is known as what? Capillary. It is single cell thick or single cell thin capillary. Diameter of this capillary is narrower than the diameter of RBC. Is it clear? If this is your RBC, you can see here why I have drawn RBC size greater than what? The capillary size. So if the RBC has to move inside this capillary, then it has to change its shape. It has to squeeze itself to get accommodated in the tiny capillaries. And this is possible because of what? Biconcave nature of RBC, that provides flexibility to squeeze itself and then get accommodated in the narrow capillaries and then can make its way forward. Is it clear? So what is the reason our nucleus is absent in RBC? To accommodate hemoglobin for oxygen carrying capacity. What is the reason RBC is biconcave? To provide flexibility, to change its shape, to move forward in the tiny capillaries. Now write the term anemia. Anemia means deficiency of what? Hemoglobin. Deficiency of hemoglobin or deficiency of RBC. That is known as anemia. So this is called anemia. How anemia will affect your body? This is going to hamper oxygen transport in the body. See, oxygen is transported in combination with hemoglobin. If the concentration of hemoglobin is lower, then this is going to reduce transportation of oxygen. And what is the role of oxygen? Why oxygen is important for our body? To convert food into energy. To convert what? Food into energy. So oxygen entering liver cell, kidney cell, everywhere, it will convert the food into glucose into energy, which is needed for the survival of the body cells. So this survival energy production will get hampered, blocked, if you are blocking oxygen transportation, reducing oxygen transportation. So that will cause weakness in the individual because of lower hemoglobin, lower oxygen carrying capacity. Now, the reasons for anemia could be multiple. So this could be due to nutritional deficiency. So nutritional deficiency like of iron, this can cause anemia. Protein malnutrition also can cause anemia. Vitamin B complex deficiency also can cause anemia. So there are several reasons for anemia. Nutritional deficiency of iron can cause anemia. Protein malnutrition can cause anemia. B complex deficiency can cause anemia. For example, deficiency of pyridoxine B6, deficiency of B9 folic acid, deficiency of cyanocobalamin B12. Either all or any one of them, deficiency can cause anemia. So there are multiple reasons for anemia, not one. But which is the most common reason in India? Iron deficiency. The most common reason in India for anemia is iron deficiency, and that also mainly in the females. 50% females in India, they have anemia associated with the iron deficiency. Anemia can be genetic also. Anemia can be genetic also. Example, sickle cell anemia. Example, sickle cell anemia. Yes, thalassemia also. We'll discuss later on that what exactly is sickle cell anemia. So this is a genetic disease. Where RBC dies in the spleen. Destruction of RBC is in the Spleen. Spleen is the graveyard of RBC. Yes. So first you write down, we will explain one by one. Have written the first 2 points. Bacteria as extracellular microbe, and the definition of virus is defined as an obligate intracellular parasite. This is the definition of a virus. Then second one is what? Self multiplication, and here we are writing what? Always needs a host cell to replicate or multiply. Third point you are writing on respiration. Both aerobic and anaerobic respiration. Now, whatever respiration we can write for virus. We have written for bacteria, it could be either aerobic in presence of oxygen or anaerobic in absence of oxygen. Whatever respiration we write for a virus. Yes. Yes. Respiration is the process of energy production. Just now today, as I told you, converting food into energy. So respiration will convert food into energy. So respiration provides survival energy. Is it clear? Respiration provides what? Survival energy for a living organism. We'll come one by one. Is it clear? First, let me write the points, then we'll explain from point number 1. So for virus, we are writing it derives energy from the respiration of its host cell. So there is no respiration in a virus. Is it clear? Neither aerobic nor anaerobic. Is it clear? So virus deriving energy from the respiration of its host cell to perform its life processes or to propagate itself. So for all the life activities, the virus will derive energy from where? The host cell. Now this host cell that you have written, host cell, this host cell can be a plant cell, it could be animal cell, it could be a bacterium, or it could be a fungus. Any one of them can act as a host for the virus. So what are the different host cell that can support a virus, support the life activities of a virus? This could be a question in prelims exam, to pick what are those host cell that can support a virus, that can support life processes, life activities of a virus. Any one of them. It could be plant cell, it could be animal cell, it could be bacterial cell, it could be a fungal cell also. Protozoa will come under animal cell only. Is it clear? This animal cell includes human body cell, It includes the birds, the different animals. It includes the amoeba, protozoa also. Nucleus? No, there are no nucleus in a virus. I will tell you now the virus structure and properties also. So what you do, you leave the space where you are writing the differences. Is it clear? There are more points to be written here. And write in a fresh page now by leaving the page here. So we can see infected host cell inside which the virus is replicating. Replication means multiplication. So we discussed just now that the host cell that can support the virus multiplication. This host cell could be a plant cell, animal cell, bacterial cell, or it could be a fungal cell also, any one of them. But what you have to remember further is that the host cell supporting the virus replication, it follows a property of host specificity. Follows a property called what? Host specificity. Host specificity means that virus adaptation to a specific host cell. Is it clear? So every virus has adapted to a specific host cell. For example, lumpy skin disease. Lumpy skin disease caused by a virus. That virus multiplies where? In the cattle, in the animal. But it does not multiply in the human body, isn't it? So virus, they have specific host. Now, so there is adaptation of virus to a specific host. There is adaptation of virus to a specific host to which it will attach, enter, and multiply. So virus attachment, penetration, multiplication into a host cell. That is known as what? Host specificity. It is not that the same virus is multiplying in plant cell, animal cell, bacterial cell. No. Virus follows a property called host specificity. So this means there is something over the host cell that the virus can recognize, to attach, to stick to the host cell. So virus is first sticking to the host cell. So to stick to the host cell, there must be some identity of the host cell, which the virus is able to identify. So there is some biological identity of the host cell that virus is able to differentiate, to which it is attaching, entering, and then multiplying. We'll take the example of COVID-19 virus to explain this. We'll take the example of what? COVID-19 virus to explain this phenomenon. Because this virus attaches, sticks to what? Human body cell, like human lung cell. So this is the SARS-CoV-2 virus responsible for COVID-19 pandemic. Now this virus has the presence of what? Spike protein. So this crown-like spikes projection that you are looking into, known as what? Spike protein. So we have seen human lung cell. Human lung cell, they are characterized by the presence of ACE2 receptor. ACE2 receptor. This ACE2 receptor helps in sticking the virus. They helps in what? Sticking the virus. Which virus? SARS-CoV-2 virus. So spike protein of SARS-CoV-2 virus is able to attach to the ACE2 receptor. And this facilitates attachment and entry of the virus into the human lung cell, and thereby enables spread of viral disease. Now, is it that ACE2 receptor is present only to attract the virus? What is the role of ACE2 receptor naturally in your body? First of all, ACE2 receptor are not only present over the lung cells, they are also present over the heart muscle cell, also present in your kidney cells also, ACE2 receptor. Their biological role in human body is to maintain the blood pressure. Maintain what? Blood pressure. But unfortunately, they also enable the stickiness of SARS-CoV-2 virus and entry and infection of SARS-CoV-2 virus. So ACE2 receptor, basically they act as gateway for the entry of the virus. Gateway for the entry of the virus. So just now we have discussed what? Host specificity. So this SARS-CoV-2 virus specificity is based on what? ACE2 receptor, which enables the virus to stick to the human lung cell, attaching, then entering, and then virus multiplies. Now, when you compare, for example, when you compare the COVID-19 wave of last year only, 2021, in the month of April, May in India, number of severe cases, hospitalization, death were the adults, not in case of the children. Is it clear? So you'll find this phenomena was more concentrated in where? In the adults, not in the children. Adults were getting more and more hospitalized and reporting deaths, not the children. What was the reason? Is it that children, they have better immunity, higher immunity? And these are related to what? So more the number of ACE2 receptor, greater intensity, the stickiness of virus to the lung cell, isn't it? So ACE2 receptor are lesser in number, their concentration are lesser in the lungs of children than the adults. This was the reason. COVID wave was, it was involving severity of case deaths in the adults, not in the children. Is it clear? So lesser number of ACE2 receptor was a blessing in disguise for children, that the pandemic involved more number of deaths in the adults rather than in the children. Yes. Antigen means the surface protein. Antigen means what? Surface protein. This antigen provides biological identity to a living organism. So surface protein is also known as what? Antigen. So this antigen allows the virus to attach to what? Human lung cell. Which antigen? Spike protein. So spike protein, it has the receptor binding domain to attach to the ACE2 receptor of human lung cell. So this is the relevance. This is the relationship between what? ACE2 receptor and the virus. So to explain you to understand, what do you mean by host specificity? What do you mean by what? Host specificity. So wherever the host has ACE2 receptor, virus has already adapted to attach, enter and multiply. Is it clear? So how virus is able to identify and land over the right landing site? So landing site of the virus is what? The ACE2 receptor of the human lung cell. Now, let me first complete. Those people who are vaccinated, through the COVID-19 vaccine, any vaccine of COVID-19. Now, this vaccine, when administered to the body, it triggers immune response. It triggers what? Immune response. We already discussed that the role of white blood cell is what? Immunity. They are the ones who protect our body against the infections or diseases. So when we are writing to trigger immune response, this will involve the white blood cells. Involve what? White blood cells. Now these white blood cells, they come into contact with the COVID-19 vaccine. For example, today we discuss what? Covaxin, isn't it? And you have written Covaxin contains inactivated SARS-CoV-2 virus. So in the process of vaccine, what is injected into your body? We are injecting the vaccine virus. Injecting what? Vaccine virus. So vaccine virus carries its antigen, carries its spike protein into the vaccinated body. So vaccine virus will carry its antigen to the vaccinated body, and vaccinated body will trigger immune response. In the immune response, white blood cells are participating, and these white blood cells, they transform into antigen specific white blood cells, which we simply call as what? Memory cells. Memory cells. So those who are vaccinated, the COVID-19 vaccine will help generate antigen specific white blood cells known as what? Memory cells. So these memory cells, they have learned the identity of the virus. Identity of the virus means antigen of the virus. An antigen of which virus? SARS-CoV-2 virus, which is the spike protein. Now, after vaccination, if the real virus enters the body, suppose this is your real virus that entered the vaccinated body. So suppose this is so after vaccination now, the real virus, the real SARS-CoV-2 virus enters the vaccinated body. How the vaccinated body will act to protect the body? This real virus entry, this will activate real virus entry. This is going to activate what? Memory cells. Memory cells in the vaccinated body. Is it clear? So these memory cells have been established through vaccine, and these memory cells now will produce anti-SARS-CoV-2 antibody. These memory cells produce what? Anti-SARS-CoV-2 antibody. This antibody we are representing by the symbol Y. So once the person is vaccinated, and after vaccination, the real virus enters the body, against which the body was vaccinated. It will trigger immune response in the memory cells. So those white blood cells that had developed memory of the antigen of the virus, memory of the spike protein of the virus, they will immediately produce in huge concentration the specific antibodies to target the virus, known as what? Anti-SARS-CoV-2 antibody, represented by the symbol Y. Now, these antibody produced by the body, how do they help us? How do they protect us? So you can see here now, these are the antibody, isn't it? These are the anti-SARS-CoV-2 antibody. These anti-SARS-CoV-2 antibody, they will block the virus attachment or infection. They will block virus attachment or infection. So these antibodies are blocking the spike protein. Is it clear? They will not allow the spike protein to interact with what? ST2 receptor in the vaccinated body. So spike protein when enters has to attach to the ST2 receptor. If person is vaccinated, the vaccinated body triggers production of antibody. And how this antibody protects you? Antibody protects you by blocking the virus attachment to the human lung cell. Is it clear? So this will be known as what? Antibody mediated immunity. This will be known as what? Antibody mediated immunity. Which is protecting us against the SARS-CoV-2 virus, provided we are vaccinated. Have you understood relationship between the virus, the host, the virus specificity, virus gateway for the entry, and finally how the vaccinated body plays a role here to protect us? Yes. Then you will not be able to see if I'm going to shrink it. Yes.
You said that the H2 receptor was present in heart lung sector.
Yes.
Is that the reason that after one person suffers from COVID, there are some heart complications also?
Yes, definitely. Long COVID involving the diabetes, also involving the heart complications, can be related to this.
But there are some reasons like after taking the medicine of RNA or mRNA medicine, people have been successfully recovered from paralysis, paraplegia, and so on. What is that?
People have recovered from?
Paralysis, paraplegia. There are some cases in the after taking the vaccination of second or third dose. For the first test, they are feeling high pain in the body.
Now, any vaccine that introduced in your body, there can be some immune response in the body because it is a foreign antigen. If you are using needle to injure your muscle fiber, there could be redness, swelling, isn't it? Pain, mild fever also. This means vaccine is working in the body.
But they are actually recovering from some other disease also, like from paralysis or paraplegia.
Recovering from other disease. In what context?
The last year, like a few patients have after taking the second
If there is some similarity of antigen, so then it can give you dual protection. Is it clear? That is the context. What is the difference between vaccine by original virus? See, vaccine by original virus, when enters your body, starts multiplying, isn't it? The vaccine virus not. So vaccine virus will be safer. In the development of vaccine, we have already ensured safety of the vaccine, that it should trigger only immune response without harming the body. Your body should develop what? Immunity. There are different methods of preparation of vaccine. This is a simple example how the antibody protects you and how it is related to the vaccine and virus receptor. We have just begun the discussion. Is it clear? We have to discuss, we have to go a long way in this discussion. See, ACE2 receptors are naturally present over your body cells, as I told you, to maintain the blood pressure. They are naturally present in your body. Yes, this is ACE2 receptor only. Now, what you have to understand is, COVID virus attaches to the body cell using which receptor? ACE2 receptor. If it is HIV virus, then it will be different. Is it clear? The gateway will be different. Coming back to our discussion now. So, why virus is known as obligate intracellular parasite? First of all, parasite means an organism dependent on another organism, either for energy, reproduction, or any other life activity. The dependent living organism will be known as what? A parasite. Now, can a virus outside the host cell, can it perform its life activities? No. So, virus remains inactive outside the host cell, but when enters the host cell becomes active. So, it is obligatory, compulsory for any virus to perform its life activities, life processes. It must be inside a particular host cell. It is obligatory. So, that is the reason why we are writing this definition for a virus, that virus is an obligate intracellular. Intracellular means inside the host cell. So, therefore, virus is defined as, so all viruses are what? Parasite. Is it a correct statement? All viruses are parasite. Yes, it is a correct statement. All viruses are parasite, but all parasites are not viruses. A parasite could be a protozoa also, like in malaria, Plasmodium. So, this is the definition of a virus, that all viruses are parasite. It is obligatory, compulsory for any virus to select a host cell, to multiply, to perform its life processes. Then, bacteria doesn't need any host cell. It performs its life activities outside the host cell. So, it is what? Extracellular microbe. Microbe means microorganism. So, outside the host cell, extracellular. Now, bacteria can multiply independently on its own, doesn't require any host cell support, but virus always requires a host cell to multiply or to replicate. So, therefore, we have written always needs a host cell to replicate or multiply. Now, but this host cell is what? Specific, based on host specificity of the virus, isn't it? The third point, respiration. Respiration is the process of producing survival energy, converting food into energy, essential for any living organism to survive. Now, this survival energy can be produced mainly in what? Aerobic respiration, in presence of oxygen. But bacteria can even undergo respiration in absence of oxygen. That is called anaerobic respiration. However, for a virus, it is neither aerobic nor anaerobic. The answer will be what? Virus derives energy from the respiration of its host cell to perform its life processes. Now, the third important point you have to write is, yes, fourth point. Culture medium or growth medium. Now, when we are using the term culture or growth medium, which means to study this organism in the lab, how lab studies are being conducted. So, if you want to gather the pure data, You have to take that organism into lab conditions. Then you have to see how it is multiplying, how much time it takes to multiply, what are the environmental conditions required for its multiplication. All these study in what? The lab condition. So in lab condition, the bacteria requires what? Culture medium or growth medium. And this culture or growth medium is a synthetic or artificial medium, also known as non-living medium. You have to provide some energy source to the bacteria. Is it clear? The food source to the bacteria. So bacteria can convert food into energy. Is it clear? So for example, nutrient agar. So this is a very common growth medium for a bacteria, to study bacteria in the lab. So you can grow bacterial colonies by giving them what? The nutrient agar as their food medium to convert food into energy, and then they can grow and multiply. Agar is a polysaccharide agar is a polysaccharide, under bracket write complex carbohydrate, under bracket write complex carbohydrate obtained from red algae. So agar is a polysaccharide under bracket complex carbohydrate obtained from red algae known as Gelidium gracilaria. So virus always needs what? A living medium. Is it clear? Virus always needs living medium. Virus can never be cultured in any non-living medium or synthetic medium. Is it clear? You can culture a bacteria in what? Non-living medium. Non-living medium also known as what? Synthetic medium or artificial medium. We cannot use any artificial medium to study virus in the lab. It has to be a living medium. And that living medium used in the lab to study the virus, it could be any one of them, as you can see here. The first one is lab animal. The most common lab animal is what? White mice, albino mice, isn't it? It could be other lab animals also, like guinea pig, rabbits also, monkey also. For example, when you are studying the vaccine virus, so vaccine virus you are injecting first in the lab animal, preclinical trial in vaccine development, and then go for what? Human trial, human clinical trial. So to study a virus, you always need what? A living host cell. It cannot be a non-living medium. And that living medium could be any one of them. What is the second one? Embryonated egg. Example, hen's egg. So inside the egg is what? Living embryo. Inside the egg is living embryo, where you are inoculating the virus, putting the virus into this embryo. But in both the case, there is what? The virus may cause death of the lab animal, death of this embryo. So this is the ethical concern in the use of whether embryonated egg or the lab animal. So therefore, the most common method is what? The cell lines. So whether it is lab animal or embryonated egg, there are ethical concerns, ethical issues or concerns, that in the process you may kill a living, whether a living embryo or the lab animal. Therefore, these are not the common method to culture the virus in the lab. The common method is what? Cell culture, tissue culture medium. So cell lines means these are group of cells derived from a specific cell or organ So these are group of cells derived from the cell division of a specific cell or organ. An example will be monkey kidney cell lines, human kidney cell lines, so basically all of you heard at least the term biopsy, isn't it? Have you heard the term biopsy? So in biopsy, what is the procedure that is adopted? You are extracting a living cell, isn't it, for diagnosis. Suppose a person is suspected of some cancer or other disease. As a confirmatory diagnosis, you have to go for what? Biopsy of that part of the body, whether lung biopsy, liver biopsy. That biopsy, you are extracting a living tissue. In the process, there is no harm to the individual. It is not the individual will die. It will in fact help us to come to the accurate diagnosis and enabling right line of treatment to save life. So similarly, these cell lines derived will not cause death of that organism. In fact, it will help in culturing the virus, to study the virus. So there are two things. One is you can study the virus. When you are putting the virus into these cell lines, it could be liver cell line, kidney cell line. Suppose it is hepatitis virus, you are testing antiviral drugs against hepatitis virus. So it will be highly drug specific also. You have to test antiviral drugs also in the lab. So antiviral drugs could be rightly tested using what? Cell lines, without causing death of any living organism, without causing any health concerns, without causing any ethical concerns. Therefore, this is the common method to culture virus in the lab. So there are 2 points. One is virus, you can write, virus cannot be cultured or grown in a synthetic or non-living medium. Virus cannot be cultured or grown in any, it cannot be grown in any synthetic or non-living medium. But it can be adopted for what? Bacteria and fungus. But it can be adopted for bacteria and the fungus. So synthetic medium for bacteria, I have already written what? Nutrient agar. Isn't it? For fungus, you can write the example of what? Potato dextrose agar. So example of synthetic or non-living medium. For bacteria, already you have written nutrient agar. For fungus, you can write potato dextrose agar. Now, dextrose is similar to glucose. Similar to what? Glucose, which we obtain from corn or the maize. We obtain from corn or the maize, the dextrose. It is similar in structure to the glucose. So how many differences you have written? 4. Just will complete this as the 5th difference also now. Genetic material. Always DNA. So in bacteria, I will write genetic material DNA. For a virus, genetic material this could be either DNA or the RNA. So for bacteria, genetic material is always DNA. Is it clear? For virus, right, genetic material could be DNA or the RNA. Some examples also we can write. Now for RNA viruses, there are multiple examples. For example, we discuss about the SARS-CoV-2 virus, that is the coronavirus. Then second example under RNA virus, right, influenza virus. Third example, right, dengue virus. Fourth, right, Nipah virus. Fifth, right, Zika virus. Now for DNA virus example, you can write DNA virus. You can see a typical virus structure for a DNA virus. So protein coat called capsid, and this capsid is protecting what? Genetic material DNA of the virus. So first example, right, adenovirus. Adenovirus. Second example, right, can you tell me other examples of DNA virus? No, it is not HIV. See, hepatitis, today I told you A, B, C, D, E. It is? Yes. So except hepatitis B, all other hepatitis viruses are RNA virus. Except hepatitis B, all other hepatitis viruses are RNA virus. Third example. Now, example also is very important. Questions will come from example and the detail. Therefore, I am writing this example to communicate you what are the important topics for the exam simultaneously. Human papilloma virus, HPV. Monkeypox virus. So this also comes under DNA virus, because this has been in current affairs. News has been repeated. So at least you should know it is what? A zoonotic virus, DNA virus. These are basic facts for the exam. I will write about HIV also now. Now HIV comes under RNA virus only, but it is what? A special category known as retrovirus. So HIV RNA, it comes under special category of RNA virus called retrovirus. So retrovirus means which can undergo reverse transcription. So this we'll discuss as the time comes to discuss on HIV. At this point of time, we remember HIV RNA is the genetic material. It comes under special category of which virus? Retrovirus, that can undergo what step? Reverse transcription. So AIDS. So advanced stage of HIV infection is what? AIDS. So question can be AIDS is a retrovirus, HIV is a retrovirus. Any statement, both statements are correct. So this is just identification of the topic. Is it clear? We have to go still a long way in discussion of these topics. We'll continue the next class from here.